The overall goal of the following experiment is to visualize the distribution and stoichiometry of epidermal growth factor receptors, or EGFRs, in the plasma membrane of intact and hydrated cells with fluorescence and electron microscopy. This is achieved by first growing EGFR-expressing cells, such as COS7, on microchips with a 50 nanometer thin silicon nitride membrane spanning over a centrally located window area. Next, the cells are incubated with a biotinylated version of the epidermal growth factor that binds to the receptor and changes the conformation of EGFR, leading to dimerization and activation. Next, the cells are fixed in order to immobilize the receptors and incubated with streptavidin conjugated quantum dots that bind to the EGF-activated receptors, and the cells are imaged. Finally, a second fixation with glutaraldehyde cross-links the streptavidin conjugated quantum dots with the EGFRs and stabilizes the sample for electron microscopy. The results show emission of red fluorescence through fluorescent quantum dot imaging and EGFR distribution and stoichiometric state or clustering via scanning transmission electron microscopy.
This method provides unique information about the stoichiometry and distribution of membrane receptors in intact cells, and can be used to obtain insight into the activation of the epidermal growth factor receptor in normal and in cancer cells. It can also be applied to study other membrane receptors, such as ion channels.
To begin, while working under a chemical hood for the first preparation steps of silicon nitride membrane microchips, and after submerging in acetone according to the text protocol, transfer the microchips directly into